Good morning to all. Welcome to the Diamond Group of Institutions, Neet and K-Cet online classes. Today's class is my molecular base of inheritance. In that, some of the previous and the concept-oriented questions are some we have discussed. Those select the correct statement. So they're given as a scientist names and some of the options are given. Out of these, some we have to identify the correct statements. So those are option A, Franklin Stalls coined the term of linkage. Here we have to identify the correct statement. See now, option A, Franklin Stalls coined the term of linkage. So in the previous class we have discussed as a plant genetics. In the plant genetics about the linkage we are going to discuss. That concept of the linkage or the term linkage was given by T.H. Morgan and Franklin and Stalls. They are not given the term of linkage. And option B, Punnett square was developed by a British scientist. Punnett square was developed by a British scientist. Yes. So the graphical representation of the alleles of the organisms in the form of a checkerboard or the diagrammatic manner that is developed by the British genetist. His name as a Punnett. So based upon his name only, we are going to represent as it is a Punnett square. So option B is the correct statement. And option C, spliceosomes states part translation. So spliceosomes state part translation. Translation mechanism means synthesis of protein. So here spliceosomes are splicing mechanism we are seen into the transcription mechanism, that is we are seen mostly in eukaryotic organisms. So here after the synthesis of mRNA is contained as a introns, exons, introns and exons. Exons are the coding sequences, introns are the non-coding sequences. For the removal of introns and addition of exons by the process of splicing will take place. So here a specialized mechanism is involved for the removing of that is introns, the removing of the introns. That mechanism is called as a spliceosome. Here specialized proteins or the enzymes will be going to bind on the surface of the introns to remove the introns. This complex mechanism, by the removing of the complex mechanism by the involvement of enzyme of introns, it's called as a spliceosome complex. Or here removal of introns from the exon, that mechanism is called as a splicing. So in the splicing mechanism, spliceosome complex or spliceosome enzyme mediated process will take place. So they given as a spliceosomes are takes a part in translation. It's not a part of a translation, it's a part of transcription of eukaryotes. And transduction was discovered by S. Altman. So here transduction was discovered by Zinder and Ledenberg. So transduction represent as it's a type of sexual reproduction in bacteria. So here transduction it is represented or discovered by the Zinder and Ledenberg, not as a Altman. So out of the four options, correct statement is B. The Punnett square was developed by the British scientist. Punnett is the scientist of British. He's a famous genetist. So here graphical representation of these alleles and organisms will be going to maintain by or discovered by the Punnett. So based upon his name, we are going to consider as a Punnett. Answer is B. And the next question, the experimental proof for semi-conservative replication of DNA was first shown in. Yes, experimental proof of the semi-conservative mode of DNA replication. So semi-conservative mode of DNA replication was explained by Messelson and Stalls. So Messelson and Stalls, they done the experiment, or it's a experimental proof for the semi-conservative mode of DNA replication. You know that semi-conservative mode of the DNA replication, you can take into any fragment of the DNA. Out of the two strands, one of the strand it came from parent, and other one is newly synthesized. That is called as a such type of the synthesis of new strands from the parents, and out of the two strands, one it is taken from the parent and other one is newly synthesized. That mechanism is called as semi-conservative mode of DNA replication. So it is seen or it was explained by the Messels and Stalls in a Escherichia coli bacteria. So they given as a fungus, bacterium, plant, and viruses. So that semi-conservative mode of the DNA replication experimentally proven by Messels and Stalls in a E. coli bacteria, Escherichia coli bacteria. Answer is B. And next question. Select the correct match. Option A, ribozyme, nucleic acids. Option B, F2 cross with the recessive parent, they given as a dihybrid cross. TH Morgan transduction and Mendel transformation. See now here, we have to identify the correct statement, means correct match. So Mendel we know they given as a match as a transformation. So transformation was done by Griffith, but they given as a Mendel is an incorrect match. And TH Morgan they given as a transduction. Transduction was done by the Ledenberg and Zinder, but here they given as a TH Morgan. TH Morgan was identified the concept of linkage. And F2 cross with the F2 generation cross with the recessive parent. F2 generation cross with the recessive parent. So to determine the genotype of the unknown organism. So they given as a dihybrid cross. Dihybrid cross means two contrasting characters are there involved. So option B also incorrect and option C also incorrect. And next, ribozyme. So ribozyme, it is a they match as nucleic acids. Ribozyme is an enzyme, and here it's a non-proteinaceous enzyme. It is a nucleic acid. So these are the ribozyme. It's seen into the part of rRNA. So the function of this ribozyme enzyme, it is involved into the transformation and also involved in the formation of the peptide bond in between the two amino acids at the time of translation. So correct statement is ribozyme. And the next question, many ribosomes may associate with a single mRNA to form multiple copies of polypeptide simultaneously. Such strings of ribosomes are termed. So here, many ribosomes may associated with a single mRNA. This is a single mRNA. So many number of the ribosomes are there arranged. Many number of the ribosomes are attached on a single mRNA to form a multiple copies of the polypeptide simultaneously. Polypeptide chain means we call as a protein only. So simultaneously synthesis of the protein is take place. That is called as options of polysome and polyhedral bodies, plastidome, nucleosomes. So when we seen as the structures here, option A, polysome. We are also called as it's a polyribosome. Polyribosomes means many number of the ribosomes are there attached on the surface of single mRNA to the synthesis of many number of the proteins. Or synthesis of the proteins are take place from the single mRNA. It's the character of prokaryotes. So the prokaryotes are they cannot perform the splicing mechanism. So that's why they can synthesize the multiple copies of proteins from the single mRNA. So that's why that is called as a polysome or polyribosome. So it is a character of prokaryotic organisms. Answer A. Next question. During DNA replication, Okazaki fragments are used to elongate. See now, in a semi-conservative mode of DNA replication, it will going to form as a Y-shaped generation. It's called as replication fork. So in these are the replication fork. One of the strand continuously synthesis of the DNA will take place. That is always DNA synthesized from the 5 prime to yes, next question. During the DNA replication, Okazaki fragments are used to elongate. So here in the DNA replication, a Y-shaped junction is formed that is called as a replication fork. In that replication fork, DNA always synthesized from the 5 prime to 3 prime direction. So here the DNA will go synthesized from the 5 prime to 3 prime direction. This is a new strand, means the parental strand should be present as 5 prime to 3 prime direction. This is, and this is a 3 prime to 5 prime direction. So now you see, always DNA will be drawn to synthesizing take place from the 5 prime to 3 prime direction. Which of the strand continuously DNA it's synthesizing take place 5 prime to 3 prime direction, that is called as a leading strand. And the opposite strand, now you see here also DNA synthesizing take place from the 5 prime to 3 prime direction only, but a small stretch or a small fragments of the DNA will be drawn to formed. That is only we call as a lagging strand. Lagging strand. So in these are the lagging strand. What are the DNA fragments are formed? These DNA small fragments are we call as a Okazaki fragments. Okazaki fragments. So now that Okazaki fragments are the lagging strand towards the replication fork. Lagging strand towards the replication fork. Formation of the lagging strand away from the replication fork. This is the replication fork. Here from this direction synthesis of the Okazaki fragments are taking place. So option A they given as it towards the replication fork, but it's not towards the replication fork. It should be away from the replication fork. And option B, leading strand away from the replication fork. So this is the leading strand and this is the lagging strand. So leading strand away from the replication fork. In the leading strand, Okazaki fragments are not formed. And option C they given as lagging strand away from the replication fork. This is the lagging strand, and this is the synthesis of the fragments are take place away from the replication, means outside of the outside to the outside from the replication fork synthesis of the Okazaki fragments are take place. And option D, leading strand towards the replication fork. That leading strands are not right. Okazaki fragments are formed in a lagging strand only. That to replication away from the replication fork. So answer it is C, the lagging strand away from the replication fork. The next question, which of the following RNAs should be most abundant in animal cells? So RNAs are three types. Those are mRNA, tRNA and rRNA. So out of the three different types of the RNAs, most abundant RNA in animal cell it is. So options are given as tRNA, mRNA, miRNA and rRNA. So out of the different types of the RNAs, rRNA, mRNA and tRNA, the percentage of the RNAs in animal cells are rRNA, it will be going to present 70 to 80% of a cell. tRNA, it will be going to present 10 to 15% of a cell, and mRNA it contains as a 2 to 5% of a cell. So out of the given three types of the RNAs, so rRNA is the most abundant RNA. And then what about that miRNA? So miRNA, it is seen into the DNA fingerprinting. So these are the small microRNA. It's contained as a 20 to 25 bases of RNA. It's called as a small fragment of the RNA. It's called as a miRNA, microRNA, which all as we are seen into the DNA fingerprinting, and also the that is RNA interference concept. So out of the three types of the RNAs, most abundant RNA it is rRNA. It's called as ribosomal RNA. And the next question, identify the correct order of organization of genetic material large to small. So we have to arrange the genetic material from the large into the small. So here they given as option A, genome, chromosome, gene and nucleotide. If you are taken as a option A, genome, a single set of the organism or haploid set of the organism genetic material we call as a genome. So in this organism haploid set of the gene, it's called as a genome. And chromosome, a arm-like structures they can form or they can having a group of the nucleosomes we call as a chromosomes. And gene, a small segment of the DNA we call as a gene. in the gene nucleotide, it represents as a single set of the nitrogen base with sugar and phosphate molecule. It all as a nucleotide. They are asking now here large to small. So here genome, it is a cell. What's the amount of the genetic material is present in a DNA? That amount of the genetic material DNA in a eukaryotes, it will going to pack in the form of a chromosome. So entire in the genome represent as a haploid chromosome number of the organism. Otherwise, haploid chromosome number of the genes or DNA, it's called as a genome. So genome is the largest genetic material, and in the genome, chromosomes, group of the chromosomes are we call as a genome, and in that chromosome, so you can imagine about the structure of the chromosome. This is the arm-like structures in the chromosome. DNA or genetic material will going to packed. Genetic material will going to packed with the help of nucleosomes. So now in the chromosome, a small fragment or segment we call as a gene. It can store the genetic information, and nucleotide. So nucleotide depend as here. The genes are made up of by nitrogen bases, sugar, and phosphate molecules. Nitrogen base, sugar plus phosphate molecules we call as a nucleotide. So nucleotide is the smallest genetic material than the gene, and gene is the smallest genetic material than the chromosome, and chromosome is the smallest genetic material than the genome. That arrangement, it is take place in a ascending order from, that is based on the genetic material. It is answer A, genome and chromosome, gene, nucleotide. So this is the way of arrangement, or this is the ascending order, the genetic material from the largest into the small. Answer is A. And remaining options are we can see as the arrangement will be going to disturb, the arrangement will, it's not present in the form of ascending order, otherwise in a increasing order or from the large into small. The next question. Which one of the following is wrongly matched? Which one of the following is wrongly matched? Options are given as a transcription. Transcription, writing information from DNA to tRNA derivation. So transcription means synthesis of the mRNA or synthesis of the RNA from the DNA. So that RNA we call as mRNA, but they given as a transcription means synthesis of the formation or writing information from DNA to tRNA. It's not called as a transcription. Instead of the tRNA, it should be present as mRNA. And translation, using information in mRNA to make protein. Yes, from the mRNA to synthesis of the protein we call as a translation mechanism. Option D is the correct only. And repressor protein. So we can see into the lactoferrin concept. So in these are the lactoferrin concept. Here, regulator gene can synthesize the regulator mRNA. That regulator mRNA can produce a repressor protein. That repressor protein can bind into the operator gene. So it will be going to bind into the operator gene. Transforming of the mRNA from the promoter into the structural genes will not take place. Enzyme synthesis will stop. So option C is also correct only. And operon. Operon represents as a group of the genes. So group of the genes arranged in a sequence order, that is called as a operon. Here under the operon, they are given as a structural genes, operator, promoter, and regulator. So here regulator genes, structural genes, promoter genes, and operator genes. These all of the genes we are combined together, we call as a operon. So out of the given 4 options, B, C, D are the correct statements, correct matched. A is the incorrect statement. So in the question also they are asked as a wrongly matched. So answer is A. Yes, next question. What is not true for genetic code? So here they are asking about the genetic code incorrect statement. So option A, genetic code is nearly universal. Genetic code is degenerate. Genetic code is unambiguous. And a codon in mRNA is read in a non-contiguous fashion. So you know that properties of the genetic code we have studied. Genetic code is a nearly universal. So here genetic code is a universal, and the genetic code is a nearly universal. Some exemptions are we discussed as a mitochondrial genome. So it's a nearly universal, and it's degenerate. Yes, genetic code it is also called as a degenerate. So why we are called as a degenerate? It is one of the amino acid can code one amino acid can code more than one codon. Otherwise, they are also called as one codon code more than so one amino acid can code more than one amino acid is called as a genetic code. It's a degenerate. So genetic code is unambiguous. So genetic code we are called as unambiguous. We are also called as a specific. One codon code for the specific amino acid. These are the codons will be coded into the other organisms also same amino acids. And option D, a codon in mRNA is read in a non-contiguous fashion. So when we see into the translation mechanism, this is the mRNA. So in these are the mRNA codons, all are they are arranged in the form of UUC, UUG like this. These all of the codons are there arranged in a contiguous fashion, but they are given as a codon in mRNA read in a non-contiguous fashion. Here, at the time of the reading or translation mechanism, these are the ribosomal complex will be going to move 5' to 3' direction in a contiguous fashion on mRNA. So in that statement, it is a codon in mRNA is read in a non-contiguous fashion. It should be present in the form of contiguous fashion. And next question, which form of RNA has structural gene resembling clover leaf model? So out of the different types of the RNAs, which RNA we call as a clover leaf model? So which RNA having clover leaf model? So those are rRNA, hnRNA, mRNA, and tRNA. So rRNA we call as ribosomal RNA. Those are having a small subunits, spherical shape structures. And hnRNA, heterogeneous RNA. So which of the RNA is light as a mRNA only? hnRNA and mRNA is same, but before the mRNA or before the functional mRNA, so first to form the mRNA is called as hnRNA. After this hnRNA can undergoing a splicing mechanism in a eukaryotes that can form into a functional mRNA. hnRNA contains both introns and exons. And mRNA. mRNA and hnRNA they are present in the form of linear structures. And tRNA. tRNA only it will be going to present in the form of clover leaf model or L-shaped structures. So this is the clover leaf model of tRNA. That is, so it was present in a animal cells or a plant cells. It should be present as 10 to 15% in the entire cell. And next question, what would be happen if a gene encoding if a gene encoding a polypeptide of 50 amino acids? So 25th codon UAU is mutated with UAA. So what would be happen if a gene encoding polypeptide of 50 amino acids 50 amino acids 25th codon of UAU is mutated to UAA. So this is the mRNA. So in this mRNA, at the time of translation mechanism, mRNA is contained as a 50 codons. So up to here it's contained as a 50 codons. So these are the 50 codons are each one of the codons encode with the one amino acid. So now 50 codons are they present on mRNA. So they are this mRNA involved into the translation mechanism. So in this 25th codon means it up to here the 50th codon means here the 25th codon it was present in the form of UAU. Instead of the UAU you are going to mutated UAA. UAA is a non-coding sequence or we are also called as a stopping codon. Stopping codon UAA, UAG and UGA, these are the stopping codon. This is the first codon and this is the 50 codon and this is the 25th codon. So what happens if the 25th codon is mutated in the form of UAA instead of UAU? If that UAU it was present means this or at the time of synthesis of polypeptide chain synthesis takes place from the first codon into the up to the region of 25th. If it's not there as a UAA, it will contain as a 50 codons in mRNA. until the 49 codons. Until the 49 codon synthesis of protein will be done to take place, or synthesis of the polypeptide chain will be done to take place. Because that 50 codon will be done contained as a non-coding sequence, means stopping codon. So instead of these UAA in the region of 25th codon, instead of the UAU in the region of 25th codon, UAA is substituted, means so that 25th codon is considering as a stopping codon. Means synthesis of the protein or polypeptide chain will take place until the 24th codon. 24th codon also reading codon only, and whenever it was reaches into the 25th codon, the 25th codon is a nonsense codon, otherwise we are also called as stopping codon. So from 1 to 24 codons is coded with the amino acids. So the polypeptide chain is contained as a 24 amino acids, and if the mutation is done in the region of 25th, this of the synthesis of polypeptide chain is stopped. That polypeptide chain is contained as a 24 amino acids. So now options are: a polypeptide chain of 24 amino acids are formed. Two polypeptides of 24 and 25 amino acids will be going to form. A polypeptide of 49 amino acids will be going to form, and a polypeptide of 25 amino acids are formed. I said it is instead of the UAA, it's contained as a UAU only. That mRNA contained as a 50 codons. 49 codons will be drawn to involve into the, or 49 codons are coding to the 49 amino acids, and they can synthesize into the polypeptide chain contained as a 49 amino acids. But here mutation is take place in the region of 24th only, 25th only. So UAA is formed. So if the 25th codon UAA is formed, means that number of amino acids in a polypeptide chain we can calculate it by the form of n-1 formula. N-1 formula, number of the codons and -1 represent as a number of amino acids. So here 25 codons are there present, means that last codon, 25th codon is a stopping codon. So that's why, and the number of amino acids are there present in that. So 25 minus 1, in that we will be as a 24 only. So to the code this polypeptide chain contain as a 24 amino acids. So option A, polypeptide of 24 amino acids will be known to formed. The next question, the pneumototus experiment prove that. See now, Diplototus pneumoniae, one of the bacteria, he was taken by the Griffith in the year of 1928. Griffith, he was done into the Diplototus pneumoniae bacteria. So he based upon the Diplototus pneumoniae bacteria, what he was proved. Option A, bacteria do not reproduce sexually. So here bacteria can perform the sexual reproduction and asexual reproduction. Asexual reproduction will take place by the binary fission and multiple fission. And the sexual reproduction of the bacteria can occur by three types: transformation, transduction, and conjugation. So these are three types of the sexual reproductions that take place in bacteria. Means option A is the incorrect statement. And option B, RNA sometimes control the production of DNA and proteins. RNA never control the production of DNA and protein synthesis, because DNA is involvement take place into the control the production of proteins and DNA, because DNA is a genetic material. And RNA means that is belongs into the RNA as a genetic material. Then also in the RNA as a genetic material, that RNA is converted into the protein synthesis. At the time, these are the DNA and other enzymes will be involved into the controlling of the process of protein synthesis. And option C, DNA is a genetic material. So Griffith experiment is done search for the genetic material at the time of the year of 1928. Griffith, he was done the experiments in a Diplototus pneumoniae. He was proposed the transformation principle. Something it was transferred from the one bacteria into other bacteria. That is experimentally proven by Griffith, and that is the search for genetic material, that is DNA as a genetic material. So answer is C. The next question, in a prokaryotes, the genetic material is, so prokaryotes are Example, we are taking bacteria. These bacterial cells does not having nucleus. So in the cytoplasm of a cell, genetic material is present in the form of a freely these genetic material we call as a NAPD. So the prokaryotic genome we are going to represent as options are they given as circular DNA without histone proteins, linear DNA without histone proteins, linear DNA with histone proteins, and the circular DNA with histone proteins. These histone proteins are absent in the prokaryotic organism, so that's why we call as in the prokaryotic genome we call as progenate or naked without histone proteins. So it is present in the form of a circular or linear. That is we have you want to know. So when we take in as a prokaryotic organism, the genetic material will be not present in the form of a circular structures. It was present in the form of many number of the foldings. Two ends of the DNA will be drawn to connected together. This is the one end and this is the one of the end. That is called a circular. And what about the linear? The linear structure of the DNA it was present. It's these two ends of the DNA does not associated together. So here in the prokaryotic organisms or prokaryotes, the genetic material it was present in the form of circular without histone protein. Histone proteins are absent and the two ends of the DNA is connected together. So that's why answer is